Matambaka, also known as Bigeye Scad, is a popular source of inexpensive animal protein in the Philippines. The term is a contraction of the Tagalog words mata, which means eye, and baka, meaning cow, which translates to cow-eyed fish. Palawan and Iloilo are the leading producers of matambaka in the municipal marine fishery. In this video, we will be dissecting Bigeye Scad, which is scientifically known as Cellar chromniphthalmus. This dissection aims to help us become familiar with the external and internal organs of the featured fish. So grab a matambaka, gloves, and dissecting tools and let the dissection begin. First things first, let's familiarize ourselves with the anatomical terminologies. Look at your fish and orient yourself with its position. The dorsal part is the top side of the fish, or back, which is usually closer to the water surface. The ventral part is the bottom, or belly of the fish, which usually contains the pelvic and anal fins. The cranial part, also known as the anterior or rostral region, which is where the head and nares are located. The caudal part is also known as the posterior region, which includes the tail fin. Now that we know the directional terms, let's familiarize ourselves with the external structures of the fish. Let's start with the structures of the head. The mouth. Mouth is used to consume food. The mandible is responsible for generating force during jaw adduction. The maxilla is responsible for generating force during locomotion. The nares, similar to nostrils, except that nares are used for smelling only. The eye is used for sight, helping the fish detect colors and see short distance. The operculum is the bony flap that protects the gills from harm. It opens and closes to allow water to pass over the gills. Now that we're done with the head, let's proceed to the external anatomy of its body. The lateral line is the line which senses vibration and movements in the water, helping the fish find prey and locate predators. The dorsal fin helps the fish balance and prevents it from rolling. The caudal fin, also called the tail fin, helps propel the fish. The anal fin, which is located behind the anus, stabilizes the fish while swimming. The anus, which removes waste and extra water, is also the outlet for eggs, or milt, which is the sperm during spawning. The pelvic fin, which is located in pairs on each side below the pectoral fins, are the fins which are homologous to the hind limbs of tetrapods. The pectoral fins are located in pairs on each side. These fins help the fish move side to side and change direction. To observe the actions of the mandible and maxilla, the fish mouth should be moved by open and close motion. As observed, the mandible is the motile part of the mouth, whereas the maxilla remains stationary. As for the teeth, matambaka has small, recurved teeth in both jaws. The teeth in the upper jaw are in a narrow band that tapers towards the back, while the lower jaw has an irregular single row of teeth. Next up is the operculum. A probe was placed in the fish mouth and was gently pushed through the operculum. The operculum is a bony flap on the side of a fish head that protects and supports its gills and helps with respiration and feeding. It opens and closes to allow water to flow over the gills, which helps regulate the flow of water and allows the fish to absorb oxygen. The gill arches were removed by cutting its dorsal and ventral attachment. The gill rakes and filaments were then identified. The gill rakes are projections that separate the gill arches from each other and create spaces between each of the gill arches. The gill filaments are feathery structures that are attached to the gill arch. They are covered with small blood vessels called capillaries, where gas exchange takes place. To facilitate the exploration of the internal organs, an incision was made along the dorsal side of the fish. Upon reaching the gills, another incision was made upward behind the gills. The third incision was made on the ventral surface just forward of the anus. The process was finished by making the last incision on the caudal part of the fish. Most of the organs reside in the ventral half of the fish body. Extra care was taken not to cut too deeply, as this might destroy some of the internal organs. The cut section of the body wall was carefully lifted away. Scissors were used to remove any membranes that adhered to the body wall. At this point, the dissection was continued per organ system. First up, the digestive system. The tan-colored liver with the attached gallbladder were identified. The liver was then freed from the body to expose the esophagus and stomach. To visualize the route a piece of food would travel as it passes through the fish, the digestive system, starting from the mouth up until the anus, was traced. Next up is the reproductive system. To determine if the fish is a male or a female, its reproductive organ was located. A large, yellow ovary filled with eggs were found, which indicates that it is a female. The ovaries are connected by tubes to the urogenital opening found posterior to the anus. Next is the respiratory and excretory system. The bright white gas-filled swim bladder was located in the dorsal portion of the fish. Located on the dorsal wall of the body cavity were the kidneys. They are connected by urinary ducts to a urinary bladder from which waste pass out of the body through the urogenital opening. Last is the circulatory system. Cutting through the fish body, all anterior to where the liver was located, exposed a cavity where the heart is suspended. The blood leaving the heart goes in the gills, where it flows through the capillaries and throughout the body in various veins, capillaries, and arteries, and finally returns to the heart to be pumped throughout the body again. This marks the end of the dissection procedure. Hope you've learned something.